All right, I think we'll get going with at least introductions. So thank you everyone who have joined so far. I hope this is not the first webinar that you've joined with Ground Zero Pharmaceuticals and Regis Technologies, but if you have, welcome. We highly encourage questions and answers session at the end. So please write down any questions that you have for Dr. Siegel or Dan throughout the presentation. My name is Lauren Mages. I am the head of business development for Ground Zero Pharmaceuticals. Dr. Evan Siegel, he's our CEO, president, and head regulatory expert at Ground Zero Pharmaceuticals. We are a strategic regulatory and product development group helping companies get from non-clinical, preclinical stage all the way through marketing submission. So we have teamed up with Regis Technologies for I think this is the third webinar that we have done. Regis will be doing one more, and then we will do a Q&A session. So we encourage everyone to come with any questions that they might have that were pertaining to any of the past webinars, or any questions that you might have that have anything to do with the developments of your program or future programs. So again, highly recommend that you come to the next Regis Technologies webinar, and then the Q&A which we will have probably in Q4. So again, we have partnered with Regis Technologies. Dan, if you want to do a little bit of introduction of your company and your position within the group.
Thanks, Laura. Can you hear me?
Yes, perfect.
Good to see you again. Looking forward to the talk today. So Regis Custom Pharma, we're part of Regis Technologies. We're a US-based contract development and manufacturing organization, or CDMO, focused on the development and manufacture of new chemical entities. We offer a comprehensive range of services, including process chemistry, analytical development, analytical validation, solid-state chemistry, micronization, GMP manufacturing, validation, late-stage development, and stability. So we've been in business for 65 years. We've got a long history of supporting hundreds of programs through clinical and a number of success stories. I don't want to talk about it anymore, and I can't figure out how to get my video on. But you guys, I don't want to belabor this, and I'll look excited to get going with this webinar. So thank you.
Thank you, Dan. And I'll pass it over to Dr. Evan Siegel now with the presentation. Again, you can put in the chat to us with any questions that you have going within the presentation, or you can wait till the end. We just ask that you stay on mute during the actual presentation. So thank you very much, and we look forward to chatting with you at the end.
Thank you, Lauren and Dan. It's a pleasure to have all of you on board here. Good morning, good afternoon, good evening, wherever you may be. And I hope that this will be interesting to you. Our topic today is expanding indications under open INDs, regulatory and product development strategies, and specifically the efficient use of existing and promising new data. Some of you have been on previous webinars, and we started with getting to the IND and other similar areas of discussion. And this one is a follow-on, but we can cover really anything you'd like in the Q&A regarding getting to the IND stage. Well, product development, the regulatory perspective, is what we're all about. We believe that regulatory affairs is the beginning of the process and continues all the way through, and is really the linchpin bringing together the chemistry, manufacturing controls, the non-clinical data, the clinical data, and all of the other parts of the puzzle that go to making up a medical product development program. But regulatory affairs is the key. Without good regulatory knowledge, without the discussions with the regulators, all programs in the most highly regulated arena in this country anyway would go nowhere. So it's very important to consider that on the left of this slide, you see the individual technical areas. And to the right, you see them overlapping more like a Venn diagram. And the importance there is just that regulatory affairs is pulling together all of the key data that you generate or plan on generating into a coherent, strategic, and tactical approach to medical product development. Well, modern regulatory strategies really need to involve realistically reviewing the product and indications, assessing emerging safety and efficacy signals frequently. And here I'll make my first pitch against the founder's disease, which essentially means that those who develop the intellectual property, who come up with the concepts, who come up with the hypotheses, must realistically assess emerging signals, that is positive signals and negative signals. And where founders falter and where companies falter, particularly small companies, is when they allow the excitement of the receptor and the target and the mechanism to overshadow the realistic data that generated from biological systems. This is a very highly complex area, drug and biologic development, and it involves biologic systems. And those systems are much more variable than those that go into making up airplanes or buildings or automobiles. So the biologic generation of data often leads to disappointment. And that's okay, because sometimes you can get around it and sometimes you can't. So that assessment of emerging safety and efficacy signals frequently throughout the development program is very, very important. Meeting early and often with regulators. Instead of avoiding the regulators, you should be going to the regulators early, getting your questions answered, and essentially de-risking your program, both for the sake of your own employees and your own consultants and the key individuals who fund your programs, and that is the financiers. So meeting early and often with regulators is important. Considering new clinical approaches, phase 1 expansion cohorts. In oncology, this is done all the time. You start with a very early program, and in oncology, often in patients. And when you find something exciting in a specific subcategory of those patients, you can expand that cohort, often without stopping a clinical trial and moving into essentially a modified clinical trial. So that's a new version of the typical phase 1, phase 2, phase 3 approach to clinical development. Multiphasic protocols, having a part A and a part B, and maybe even a part C, whereby as the data are generated, you can assess those data and then make key decisions at critical points so that you can either modify or slow down or enhance that clinical trial to get the most out of it and to get the results at the end farther along in the development pathway, if the biological signals allow for this. Adaptive designs, you've all heard of those. Target and markers across tumor types in oncology. This is very important. It used to be that the origin tissue or organs of a cancer determined what type of cancer you have and the treatment selected therefore. But now you often see the same targets and biomarkers across tumor types, similar tumor types, but stemming from very different organs. So for example, breast cancer and gastrointestinal stromal tumors often have similar markers. So you can treat them similarly. So the idea of applying your new technologies and your new therapeutics to a new way of thinking about targets and biomarkers, that's a very, very important new approach to clinical development. And therefore, the modern regulatory strategies must take this into account. New indications, go, no-go decision-making. Take a look at your market. Make your realistic financial profitability projections. Assess your previous and emerging data ruthlessly. That goes back to the look at your emerging safety and efficacy signals. Consider the costs and time for development realistically. You may think that your product has a possibility of 10 or 15 indications. Small companies certainly cannot study 10 or 15 indications simultaneously, and very often large companies have to establish priorities. It's very important not to think that you're going to be a one-stop cure-all. You might eventually be a pembrolizumab, which in many ways is a one-stop cure-all for many types of oncology indications. But it's taking a long time to get there, and more than 18 to 20 approvals to have a product that is so pan-effective in so many different indications. So you need to really realistically set your priorities and kind of hold your horses in expanding your program to the point where you basically bankrupt yourselves because you don't have enough money to continue the 1, 2, or 3 most promising programs. If you're a leader in a new area, be aware of the learning curve for the FDA. New opportunities can be mixed in effect. That is, the agency may get very excited by a new mechanism of action. They also may be very cautious about safety because, as you know, there are two types of primary toxicities that occur with a drug candidate or a biologic drug candidate. One is structural or innate toxicity, whereby the molecule simply has a negative interaction with tissues. The second is pharmacodynamic toxicity, where the very binding to a marker, to a target, or the very action on an enzymatic pathway that leads to efficacy can also lead to toxicity due to the primary action of the product. So you have to be very careful to separate out your toxicities as well and make go, no-go decisions on the basis of the risk versus the benefit for each indication. If you are a follower, make sure you don't ignore known previous issues for similar products. It's very, very seductive to take a look at a product, have your own product, which is similar to a product on the market, and say, yeah, but ours is different. Ours is better. And the FDA's basic philosophy is prove it. I'm from Missouri. Prove it. Prove that you're better in safety and or better in efficacy. So the fact that you may have a product that's similar to a product that's already on the market does not mean that you have a buy on assuring the appropriate balance between safety and efficacy. And then making sure whether a new IND is necessary. There are certain situations where an IND is not necessary. If for certain bioequivalence or bioavailability studies, an IND is often not necessary. So make sure when you look at your go, no-go decision-making, your regulatory strategy, that you realistically assess your program and where it is, use appropriate consultants, and then make reasoned decisions each step along the way. A new indication may be added to an existing IND if the new indication is covered by the existing reviewing division in CDER, for example, Center for Drug Evaluation and Research. So as you probably know, there are 3 oncology reviewing divisions for solid tumors and 2 for hematologic malignancies. So if your indication, new indication under your present IND or intended under your present IND is in a different reviewing division, you may actually need to submit a new IND, mostly copying the previous IND, to the new division in which your new indication is regulated. So you have to be very careful about regulatory strategy there. If the product is still covered by the applicable CBER office, Center for Biologics Evaluation Research, so OVRR, Office of Vaccine Research and Review, or Office of Tissues and Advanced Therapies, OTAT. This is a different situation in CBER than in CDER. For vaccines, for live biotherapeutics, for cancer vaccines, for cellular therapies and gene therapies, those are all regulated in CBER. And the organization of CBER and how they break up therapeutic areas is very different than in CDER. So you just have to be aware of that. You may add to an indication to an existing IND if the product is the same structure, assuming the other provisos are fulfilled. If you've basically got the same molecule, even if you change the formulation of the route of administration, you can often add an indication to an existing IND. If the primary mode of action of a new combination product includes the original API and lies within that API, then you may actually be able to simply add a combination product to your existing IND in the same reviewing division in CDER or the same reviewing office in CBER. And if the route of administration, unit dose, dosing frequency, and or targeted patient population are the only changes for the new indication, you may often add a new indication to an existing IND. So why is this important? Well, obviously, even if your IND is mostly copied and electronically submitted to a new reviewing division, it still takes some time and money to do it. And you have to carefully QA and QC your new IND. And it's just a little bit more time and resource and financially intensive than adding an indication to your present IND, although you still have to justify that new indication based on pharmacodynamic activity, assurance of continued safety under the new indication for that same product that's already under the IND, et cetera. A complete new IND may be required or beneficial if the new indication is in a different reviewing division. As I mentioned earlier, the product is not covered by the applicable CBER office for certain biologic therapies. If the original IND is not in electronic format. Now that's very important. If you have an old IND and you want to submit basically a new indication of that old IND, you may actually need to submit an entire new electronic IND. All commercial INDs must be submitted electronically with very specific format guidelines and electronic standards, electronic submission standards. So essentially an investigator IND, an investigator-sponsored trial, does not need to go electronically, although we still recommend it. But all commercial INDs by commercial sponsors do need to be submitted electronically. So you actually may need a new IND if you're submitting a significant new indication to an old IND that is not presently in electronic format. And that's at the behest of the reviewing division. You have to contact the regulatory program manager and see if they will still accept information on the new indication in either paper format or electronically, but keeping the paper formatted IND still active in that division. And if future corporate plans include out-licensing or selling individual indications, this is very important. We have been approached no fewer than 6 or 7 times in the last years for a new indication to an existing IND, but there's an intent by the sponsor to out-license individual indications to other parties. Well, under those circumstances, you'd have to have right of reference to the entire IND package given to all of these new entities that will own indications. And all of the safety information has to be cross-referenced across all the INDs for that molecule. So it becomes very unwieldy and very difficult. And if a sponsor goes bankrupt and that sponsor's IND becomes inactive or becomes removed by the FDA, no one can reference it anymore. So it may be beneficial for a sponsor of a new indication to submit a complete new IND. New protocols versus new INDs. What do you need for clinical trial initiation for new indications? As you know, in the United States, an IND is a dossier for a specific product. The formulation can change, but the API needs to stay the same. And then you have a product dossier that keeps being built upon with new protocols and new non-clinical information, new chemistry, manufacturing controls information. In Europe, you have a CTA, which is a clinical trial specific dossier. It's not a program dossier. You can't add new clinical trials. For every CTA, for every clinical trial, you have to create a new CTA. Same thing in Australia. Every clinical trial has either a CTN, clinical trial notification, or CTA, a clinical trial authorization. What happens then is that there's no central dossier the way there is in the US. The good news in the US is you can keep adding and adding and adding to that dossier and essentially create a mock or an early marketing application by building up your IND the way you're supposed to. So CMC data. If there's a formulation change, new analytical data, new stability data, and updating of DMFs need to be need to be submitted. You may need to bridge GLP toxicology studies. If you're changing the route of administration and your initial tox package does not cover that route of administration, then you may actually need a small bridging tox study to get into the new indication, because it's got a different route of administration. Therefore, it has a new safety profile that may be attendant to it. And ADME and PK as needed, because if there's a formulation change, you may need some new ADME and PK to move ahead. The preclinical, non-clinical package for new indications to INDs would involve pharmacodynamic studies, in vitro and or animal models. So you can't just say, well, we're going to try this new indication without some proof that that new indication is viable for your product. You may possibly need new GLP tox studies in 1 or 2 species to cover the duration of the human trial or new patient cohorts. So we've seen this happen before. Let's say that you have basically an anti-inflammatory product, which is effective in an oncology indication. Because as you know, there is a strong connection between inflammation and metastasis in certain oncology indications. So you have a powerful anti-inflammatory. It's proven effective in certain oncology indications. Guess what? You want to study it in rheumatoid arthritis. Well, the risk-benefit profile of a product for rheumatoid arthritis is vastly different than that for serious or life-threatening oncology indication. So the FDA may say, and we've actually had them say this to sponsors, well, wait a minute, oncology may have allowed this, but we're not going to, because the risk is much higher against the benefit for rheumatoid arthritis patients for your product than it is in oncology. So changing the indication or adding an indication may change the risk-benefit profile, thus requiring additional submissions of tox studies, maybe even safety pharmacology if they were never submitted prior, but certainly GLP tox studies may be needed. And again, ADME and PK in animals may be needed to assure that the changes that you're making may not give you what you expect to get. So your ADME and PK may need to be enhanced. Other preclinical studies. You may need developmental and reproductive toxicity studies if, again, the risk-benefit profile changes, say, away from an oncology indication to rheumatoid arthritis or psoriasis or atopic dermatitis of something of that nature, which are definitely less serious or life-threatening than most oncology indications. Irritation local tolerance for dermal or parenterally dosed products. So if you're changing your route of administration, you may actually need some local sensitivity, irritation, phototoxicity, and other studies. And then juvenile animal studies. If you're going into a pediatric indication, for some pediatric indications, going from adult to pediatrics, the FDA may impose the need to use juvenile animals to make sure that the risk profile of your juvenile population or your pediatric population is not greater, significantly greater than that for your already extant adult dossier. Something very important, creating reviewable reports from early data. You may have university laboratory or clinic early data from basically laboratory experiments, which indicate a new indication could be very, very viable for your product. Well, those data need to be in the appropriate form and format. So pharmacology reports, primary pharmacodynamics, secondary pharmacodynamics, the ratio of on and off target effects. Those data need to be in the form of reviewable reports, not lab notebooks or even electronic lab notebooks. They need to be in the form of reports. So too, safety pharmacology, tox studies, ADME, and bioanalytical methods need to be in reviewable reports. The difference between discovery and very early development work in a university or clinic laboratory compared to a more commercial program that needs to generate, in all cases, reviewable reports from the early data. So as an example, let's take a look at a pharmacology report from early laboratory data. You need a prospectively written protocol, all protocol amendments, or a list of detailed methodologies and SOPs for conducting the study. The detailed description of the study design with all of these descriptors in it. Results for all parameters evaluated for each animal on study. No cherry picking. All the good, all the bad, all the ugly need to be included in the pharmacology report from early data, and an analysis and interpretation of the study data. And what substitutes for a report in many people's minds is a published paper. And I can tell you, published papers are almost always considered secondary and supportive, not primary, for regulatory review for a new indication for an existing product, or for a first indication for a new product. So you need to generate pharmacology reports in the case of pharmacology, tox reports in the case of toxicology, ADME or PK reports in the case of those respective types of studies. So this is a big, big separation between a program intended to develop a therapeutic or an imaging agent or what have you, versus a program which is essentially intended to create and validate early on concepts, but not lead to product development. In the product development bailiwick, you need reviewable reports that regulators can review. For clinical data for a new indication, previous clinical safety data should be summarized, preferably draft or final clinical study reports from previous studies, a summary of the upcoming clinical development program for the new indication, a detailed protocol synopsis for a new study for phase 1 or 1/2, and preferably a full protocol for phase 2 or 3. And note the use of special protocol assessments for critical confirmatory studies. You may, in certain instances, have a new indication that you've been studying where you've been in other countries, for example, looking at this new indication. Now you want to come to the US and you want to do one, preferably one, maybe two confirmatory studies based on the data you have generated from the earlier studies elsewhere. Well, the best way to do that, in my opinion, is a special protocol assessment where you run the entire protocol, the statistical analysis plan, the data safety management board or the data safety board or the safety data board, whatever you want to call it, charter, put that in a package and send it to the FDA and ask for a special protocol assessment. If they agree with your program, and they often don't initially, they will have detailed comments and you're supposed to incorporate them or justify why you don't want to or don't think you need to, you eventually will hopefully get an agreement in writing from the FDA. That means that if you execute that study per protocol and the results look decent from a statistical significance point of view and a clinical relevance point of view, the FDA must consider that study as part of a dossier to be reviewed for a new drug application or a biologics license application. It doesn't mean they have to approve it, but it means that that study suffices for full review for that study as part of a marketing application. So that special protocol assessment can be very important for critical confirmatory studies. And they don't have to be phase 3. For a lot of indications in oncology, you may get an end of phase 2 approval. So you could have a large phase 2 and put it under special protocol assessment if the FDA agrees that your product is ready for a confirmatory study. And look at the concordance of the protocol with other information. The protocol should be appropriate for the stage of investigation desired for the new indication. Safety support is critical. Pharmacology data, as I mentioned earlier, should support the proposed study. And manufacturing data should allow for a thorough review of the process and quality attributes of the drug substance and drug product. And Dan can speak at length to this as necessary. So basically, you have all of your puzzle pieces appropriate for the stage of investigation where you find yourself, phase 1, 2, 3, et cetera, for the new indication. And all must harmonize. Your CMC needs to harmonize for the stage of investigation with your clinical, and all the other pieces need to be in place to be able to move ahead with the new indication. For expanding indications, a pre-IND meeting may be desirable if a new IND is necessary. Now, you can get a special meeting, and you can call it a pre-IND meeting for a new indication under an existing IND. The FDA will sometimes allow that, but typically it will be for a new IND. You can request a type C meeting, which is a kind of anything meeting, to discuss a new indication under your present IND. That's always available to you once you have an open IND, time permitting for the FDA to look at this information. But the pre-IND role in strategic development is critical. It identifies non-clinical studies that will support the initiation of new clinical trials, de-risk the IND submission, discuss available methods to enhance development, such as orphan drug designation, fast track, breakthrough, animal efficacy rule, pediatric development program, rare pediatric disease designation, all of these buzzwords that many of you are familiar with and some of you may not be. Those are the key elements, all of these elements for the role of a pre-IND meeting. And the FDA often identifies issues, CMC issues, stability, compatibility with the clinical protocol, formulation changes, and how they affect product quality. The preclinical, non-clinical data, how strong are the pharmacodynamic data for the new indication? What about the clinical protocol? You may have a good safety profile in your initial cohort, but when you go to a new cohort of patients, that risk-benefit may shift. So that's always looked at the pre-IND. And regulatory strategy, aggressiveness of the program and timing. The FDA may say, well, wait a minute, you're talking about, a 300-patient phase 2 study after a 30-patient initial study in your new indication. Let's not think about 300 patients going from 30. So again, they'll provide you advice on the rapidity of movement of the program and how that might be projected. Post-meeting events after a pre-IND consultation. Read your notes and the FDA concerns carefully. Don't ignore boilerplate, the pediatric development plan, clinical non-clinical safety reporting system. All of these boilerplates that the FDA tends to put into the consultation are important because they do apply to most programs at one time or another. Make sure you talk with your investigators about protocol changes, study participant availability, number of participants in the trial. A pre-IND consultation can de-risk the upcoming IND if it needs to be a new one for a new indication. But it can also create some roadblocks to moving ahead as quickly as your corporate timelines dictate. So all of these issues are part of the dance, the regulatory dance, the regulatory-led symphony of putting together all of the key pieces, all of the key players, and rendering your program harmonious in terms of moving forward. The IND package, I think everyone knows this on this call. You have the administrative information, the technical summaries. First is module 1. The IND technical summaries are module 2. The CMC details, module 3. Non-clinical details, module 4. And clinical detail is module 5. And ECTD and electronic submission are required for commercial INDs, as I mentioned earlier. I'm going to skip case studies in the interest of time because I think case studies will come from the Q&A, and we can do Q&A now and take as long as you all would like for it today. And thank you.
Yeah, if people want to put the questions that they have down in the Q&A box or the chat box, I'll be taking a look at that. Or if everything goes smoothly as far as people taking turns, feel free to go ahead and ask your questions.
You think, oh, I'm just not sure if you can hear me.
Yes.
Oh, yeah, sorry, it's Roger.
Hi, Roger.
I'm just I kind of wondering on the new indication, do you think it's better to try and do it within the IND or to get approval for a more narrow indication from the get-go? And then now that you have an approved product, try and get it an expanded indication?
It's an excellent question. In general, the establishment of your first indication is a very, very key strategic decision. And normally what I tell people is to target your first indication for your most promising part of your program or your most promising approach, and then worry about additional indications later. But putting it in the same IND is completely permissible, and it allows the reviewers to really be able to tap that IND for all of the supportive information that is appropriate to and attendant to your new indication. So targeting a first indication that's most promising in terms of quantity and quality of data, maybe critical unmet medical need. That's a good one to target for your first indication if you have one like that. But then putting your second indication in the same IND is not only permissible, it's probably desirable because it cuts down on the quote paperwork that the FDA has to deal with.
But if if you were, say, you had the data completed for the first one, but not completed for the second one, I don't suppose they're going to approve the other one and let the IND stay open while you continue to work through that second indication?
Well, your IND stays open. Your IND stays open anyway. So basically, when you get an approval, what I would do is I would go for an approval as quickly as you can for that first indication and keep submitting for the second indication to the same IND. And then you can do a supplemental marketing application, a BLA or an NDA for the second or third and fourth indications. But the IND stays open as an open dossier, and you can dump all of your information for the new indication into it. The IND doesn't disappear when the product gets approved, is what I'm trying to say.
Oh, okay. So because we had like the initial one is for a single dose, and the add-on would be for repetitive dosing. And so I'm assuming they would want animal studies on repetitive dosing and stuff, which we don't have yet.
Yeah, I mean, if the products are serious or life-threatening illness, and you don't have repeat dosing tox data, for example, you may actually be able to do a small pilot in the new patient cohort or in the same patient cohort very, very cautiously based on the safety data you have from the first. Normally, you'd need some bridging tox, a short-term repeat dose study, probably in one species, if it's serious life-threatening illness with an unmet medical need, to support that repeat dosing. Normally, you would need it. In rare instances, the FDA may allow you a very cautious repeat dosing at a lower dose and then move up in an escalating dose manner using a modified Fibonacci calculation to do the repeat dosing. That would be a matter for discussion with the reviewing division.
Oh, very good. That's very helpful. Thank you.
Sure. We have a question. How do you address building an analytical package for relatively new drug types, i.e. radio-labeled ADCs for CT and regs? Okay, I'm not quite sure what the latter part of that means, but the analytical package radio-labeled ADCs have a pretty long history now. And one way to do it is to simply look in approved radiopharmaceutical NDAs for what they did. That doesn't mean that you have to or should follow it extremely closely. But there are new guidances on radiolabeled radiopharmaceuticals, and ADCs have a pretty significant history. And so you can look at the most recent approval packages and get an idea for the kinds of analytical you'd need. Plus, well, like in our company, we have experts in CMC, and I'm sure Dan does as well. And they can help you determine what kind of analytical you need. You obviously have to look at radiolytic decomposition, stability, et cetera. But there's a pretty well developed series of packages of analytical methodologies for ADCs and radiolabeled ADCs at this point. I hope that helps. Clinical questions about new indications? Anyone want to have a question on that? Here we go. Would you recommend microdosing strategies for new drugs indications? Well, microdosing strategies get you in quicker without a lot of additional tox. The problem is that if you've already got an indication that you're studying, you've likely got some significant tox, and microdosing really doesn't gain you anything except some pharmacological information, maybe some ADME PK, but it doesn't give you any efficacy. I mean, unless you want to consider that microdosing is sometimes used to determine or to describe imaging agents, quote, microdoses of imaging agents, in which case you can actually get imaging data with microdosing. But from a time, resource, and financial point of view, microdosing really only gives you very early exploratory data, and won't necessarily help you with a program that's fairly far along in one indication going into a second indication. If it's a radically different patient population, you can certainly get some ADME, and you can get some target binding information out of it, but you can probably do that better without actually doing a clinical microdosing study.
If any of you have questions that might come up, that might come to mind later down the road in the next day or two, feel free to email me. Evan has my email address up there, lmagis@groundzerous.com. You can email me with any questions. If you want to get the slides to this presentation, again, shoot me an email. And if you have any questions for Dan on the CDMO side, you can reach out to me and I'll forward along your email as well. If there are some last minute questions, please speak now or send over the question in the Q&A box.
What happens if a clinical trial fails? Well, it depends. If it's a clinical trial for a new indication, it doesn't adversely affect the initial indication or indications, particularly if it has to do with efficacy. If there's a fail, quote, end quote, due to a safety concern, and you can tie it to the new patient cohort or the condition that you're trying to treat, then no harm, no foul. But if a clinical trial fails for a new indication, then that's all right. If a clinical trial fails an initial indication and you want to go to a second indication or a third indication, that's fine as well. But all of the safety data do need to be provided to the agency, which they do anyway under IND regulations periodically. Good questions. Keep them coming.
Okay, I think we'll give another minute for any questions to come through. Again, if you have anything that comes up, or if you do want to talk to us to get any idea of consulting strategy, again, clinical or preclinical. We do all ECD submissions in-house too, so feel free to reach out if you want to set up a call to learn a little bit more about that, or again, just to get some last minute questions answered. All right. Well, thank you everyone for joining. Please look out for our postings for our next webinar, which Regis Technologies will be hosting. We make sure that you follow both of our company's pages on LinkedIn and us individually for any updates. I hope that to see all of you at our next webinar, and have a great rest of your week. Oh, actually,
With
we have
one more
quick
question.
Yep.
Does the FDA look more favorably on indications for acute or more chronic disease? There's no practical difference. The FDA will look favorably on an indication for an acute or chronic disease. They will have special, there will be special significance placed on serious or life-threatening indications with an unmet medical need, because those indicate clear and present need for those patient cohorts. So I think there's no difference between acute or chronic per se. It depends on the seriousness and whether there's an unmet medical need or not. I mean, the FDA is looking at multiple products for essentially every indication you could imagine right now, and they're pretty even-handed, but they put special emphasis, and there are special programs in place to deal with serious or life-threatening illness, particularly with unmet medical need. If there's nothing on the market, or what's on the market doesn't work very, very well. So you don't have to worry if you've got a chronic disease indication that the FDA will, look unfavorably on, or to put it as a lower priority. They don't, but they do look at serious or life-threatening and with an unmet medical need. That's the highest priority, is serious or life-threatening illness with an unmet medical need.
If there are no questions, I just want to say thanks again, Evan. I think that was a great talk, and so thank you from everybody at Regis Technologies.
Thank you.
Really looking forward to getting on and having the next presentation, which is going to feature the more process chemistry and CMC.
Great. Thank you.
Absolutely. Thank you, Dan. We're looking forward to your next conversation. Have a great week, guys.
Thanks all.